MitoSENS Webinar. I believe this is the first webinar that's hosted by Leaf, or at least the first one that I'm actually part of. You can confirm or deny that, Steve. So I'm Oliver Medvedik. I'm broadcasting live as of the moment from Cooper Union, New York City. And I'm a member of Leaf, Life Extension Advocacy Foundation. And the campaigns that we launch can be seen at www.lifespan.io. And this MitoSENS webinar is going to be related to a campaign, actually one of our first campaigns that we launched very successfully a while back. And I'm just going to share my screen here with you. The MitoSENS campaign. And we're going to talk a little bit about this because this project has progressed significantly since then. So you can see it had a very successful launch. And things have been rolling along quite well since then. So I'm going to stop sharing the screen and come right back to me. With us, we have a number of different people, but included in the bunch are individuals from SENS, which is the Strategies for an Engineered Negligible Senescence organization. We have Michael Ray, Aubrey de Grey, Matthew Oki O'Connor, and Amutha Uminathan. Okay, so without much further ado, I'm going to launch off this webinar, and basically it's going to be a series of, I guess, Q&A on the topic. And I want to, for our viewers, I want to make sure that we have a kind of a grounding as to what we're going to discuss. And the MitoSENS project obviously is centered on mitochondria. So before we kind of begin, we should kind of frame the problem a little bit, a little introduction and background for everybody. And I guess we'll start really at our kind of organelle of choice here, which is the mitochondria. And I'm going to toss this over to the SENS folks. And whoever wants to basically tackle this first question, which is a little introduction into mitochondria and what they do. Obviously we can talk all day about mitochondria, starting with the Krebs cycle and so on and so forth. But I guess we'll probably want to keep it a little bit focused to relevance towards our project and aging. So what are mitochondria and what do they do?
Okay, well, mitochondria are the powerhouses of the cell, and they generate most of your energy in the form of a chemical energy form called ATP. And they are, in a way, what distinguishes us from the lowest life forms like bacteria, and in that bacteria can only generate energy using glycolysis or aerobic respiration, while complicated life forms called eukaryotes can generate energy doing both glycolysis and oxidative phosphorylation or aerobic respiration. So this form of energy generation is the reason that we breathe oxygen in order to generate this energy. So mitochondria, they live inside of our cells. There's hundreds or thousands of them per cell, and they are what allows us to efficiently generate energy to have a complicated life.
And you mentioned there's hundreds, if not thousands of them, depending on, the cell type in our bodies, and they generate energy. And I know they, a lot of our viewers here, there's a lot of other things they do, but obviously central to their function is generating ATP. But they are, evolutionarily, they used to be somewhat independent, right? And now they've kind of lost their independence, but they still have DNA, and they can still independently replicate more or less. So kind of related to your project, how does the fact that they still have a genome, albeit a small one, but an important one, how is that connected to your project, and how is that related to the aging process?
Yeah, so that's right. So evolutionarily, a protobacteria got engulfed by another cell, and then over evolution, the protobacteria transferred much of its genes to the nuclear genome, in the sense that more than 1,000 of the protein, that the proteome that makes up the mitochondria is actually synthesized in the cytosol and imported into the mitochondria. but for some reason, the organelle itself retained a small subset of genetic material called mitochondrial DNA here. And the proteins that are encoded from this mitochondrial DNA are all part of the OXPHOS relay, or the mechanisms that generate the ATP for the cell. So for some reason, it has retained within itself a small amount of DNA, but it has, given up much of the DNA. It is transferred to the nucleus, but that's how the symbiosis between the nuclear genome and the mitochondrial DNA happens. But then the OXPHOS relay itself, the byproducts of the oxidative phosphorylation is that there's a lot of reactive oxygen species created, and the mitochondrial DNA is in many aspects very different from the nuclear DNA. Of course, it doesn't have any of the protective factors that are in the nuclear DNA, like histones or other protective protein molecules that keep the DNA intact when not in replication. And also the repair mechanisms, like should there be a mutation or a deletion or an insertion, the mitochondrial DNA repair mechanism is not as advanced as the nuclear genome. So with time, the byproducts and the inefficient repair mechanism all add up, and they accumulate mutations with time. And since all these genes are very critical to the oxidative phosphorylation relay, any mutation in that per se is going to affect it very adversely.
So basically what I'm hearing is that one of the issues is that mitochondria, even though they're vital, they generate a lot of reactive oxygen species as a byproduct of their functioning. And having DNA in very close proximity to these reactions, as well as having a kind of a lack of in-house repair capabilities within the mitochondria that is much more, I guess, established within the nucleus, those two conditions kind of set the stage for a bad situation for the DNA that's inside mitochondria. Is that kind of a good summary, I guess?
Yeah.
And so how does that exactly tie into aging?
So with age, there's a bunch of changes in the mitochondria, and some of those are functional changes. They're regulated changes that are responding to different ways that the organism is aging as a whole. But the most prominent changes, and the ones that you have to be most worried about, are the ones that are actually damage to the mitochondria themselves. You see a very small number of cells in the aging organism that are completely taken over by mitochondria that have these very large deletion mutations. And the problem with deletions is there's no template that can repair them. So once they happen, they are irreversible, and the way the cell would normally deal with that is to just generate more mitochondria and get rid of those old mitochondria. And for reasons that are a little bit disputed, what happens instead is that the large deletion mutations take over the entire cell and impair that cell. And you see that very closely correlated, certainly, and in some cases, mechanistically connected to a lot of age-related dysfunction and age-related diseases. The strongest cases there are Parkinson's disease, where these kinds of cells are very important in the area that you lose a lot of cells and that thereby cause the prominent motor dysfunctions with Parkinson's disease, and also in skeletal muscle, where they happen focally, and because of the elongated structure of the muscle fiber, they lead to muscle fiber breakage, which is a major driver of sarcopenia, which is the loss of not only muscle mass but muscle function with age. So the result of all these things is a lot of age-related dysfunction and disease. It also, there's reason to think, spreads oxidative stress through the entire body, which then just causes problems of other kinds, like lipofuscin accumulation or increased cellular senescence. So it is a bad thing, and the MitoSENS project, what we call allotopic expression, is to create a solution to that problem whereby even if the mitochondrial copies of those genes are knocked out as a result of these large deletions, you can still provide the proteins that those genes encode so that the mitochondria themselves can keep functioning with age. And that just bypasses the entire issue of whether you have these deletions or not. The cell can keep functioning. You're not producing this excessive oxidative stress, and it's effectively functioning as if it was still a young functioning cell, even though you might have deletions in some of those cells.
Okay, so that's a good summary of what happens as a result of the damage and kind of the direction that the MitoSENS project has gone towards alleviating it. But just a little bit of kind of specifics in that direction. Maybe one of the authors of the paper could kind of comment a little bit. The project that we funded ended in 2016. The paper was published, and it was demonstrated that this allotopic expression worked. But how exactly was this achieved? And what I mean by that is what genes were removed. There's, I believe, about 13 genes that are still in the mitochondria. So what genes were focused, and how was this allotopic expression achieved? And kind of in a kind of a brief summary, what were the results that were promising?
Yeah, I can summarize that. So there are several. So all these thousands of genes that have migrated to the mitochondria, to the nuclear genome, already have specific features. So, they typically contain a mitochondrial targeting sequence. And another thing to remember is that the codon usage between the mitochondrial DNA and the nuclear DNA is different. So the mitochondrial DNA still uses the bacterial codon usage, whereas eukaryotes, or the nuclear DNA, uses the eukaryotic codon usage. So what we had to do was, taking all these into consideration, re-engineer. We did this to two of the complex 5 gene. Complex 5 is the ultimate complex in the OXPAS relay that is actually the ATP synthase complex. This is the ATP8 and the ATP6 gene. So what we did was we re-engineered the ATP8 and ATP6 with respect to codon usage for the nuclear context. We add an upstream mitochondrial targeting sequence, and we express them stably in a patient-derived cell line that had a mutation in the ATP8 gene. Now, this mutation is so severe in that it truncates the ATP8 protein 2/3 into it, and the complex 5 is completely disassembled. What we were able to show is that when we expressed our allotopic constructs from the nucleus, we were able to bring back the complex 5 assembly. We were also able to bring back the complex 5 function, that is the ATP synthase activity or the ATP hydrolysis activity, to about 55%. But to achieve this, we had to co-express both these proteins. In the mitochondrial DNA, the ATP8 and ATP6 genes are translated from the same mRNA. It's a bicistronic mRNA. It's, again, the remnant of a bacterial system, but we express them individually, and we were able to bring back the function significantly, and we were able to rescue these patient cell lines.
So these patient cell lines, so when you said you expressed and you rescued, I'm taking that this expression was done through a transgene that was in the nucleus?
Yes, that's exactly right. So we created vectors. So they were these vectors would have these genes individually, and we co-transfect these vectors into the nuclear genome, and we select for stable selections. So these two proteins are produced from two different locations in the nuclear DNA, and they are translated in the cytosol. They are imported through the mitochondrial import machinery, and it assembles into the complex 5 in the mitochondria. So we are hacking the mitochondrial import machinery for all the other 1,000 proteins that it's evolutionarily it's already done. They are hacking the same system and trying to achieve correct incorporation into the correct complexes through the import process.
So a lot of engineering challenges involved, moving things from the mitochondria to the nucleus, then obviously back again. So you said these were patient-derived cells. So obviously these mutations occur spontaneously in humans that are outside of what we would consider normal aging. What are some of the are some of the I guess symptoms and phenotypes of people that have these types of mutations, specifically the mitochondrial mutations that you looked at? How severe are they for individuals? What are some of the consequences? And I know, Michael, you briefly listed some other conditions that are a result of mitochondrial mutations. But can you maybe elaborate a little bit further on the ones you worked on?
So an important thing to understand is that when you get these large deletions in aging cells, the issue is not so much that you're knocking out, this particular protein coding gene or that particular protein gene, because one of the things these large deletions do is that they knock out the cell's ability or the mitochondria's ability to generate their transfer RNAs, which are the RNAs that actually take the amino acids and shuttle them over to assemble with the mitochondrial ribosomes so that they can extend out the proteins that are coded in the mRNA. So the result of that is once you get these large deletions, which again, when they occur, they rapidly take over the entire cell, it doesn't matter exactly which genes you've knocked out. The entire process of generating new proteins just shuts down. So you really have to replace them all. It's not like you see, a lot of aging people where they have a cell where, one individual gene in the mitochondria has been knocked out. If that were the case, that particular mitochondrion would just get removed by mitophagy and replaced by healthy mitochondria. It's when you get these large deletions that you get the clonal expansion and the cell really becomes dysfunctional. And at that point, you can't just go after one or two specific mutations, which would be very hard to do anyway. You really have to go through them wholesale. So we're building one by one to get the full 13.
Right, and in the one by one case, what was so Amutha, you mentioned that these came from patient-derived cells. So these weren't aged mitochondrial mutations, but these were a mutation that is I guess congenital. I'm not exactly certain how it arose.
No, yeah, we received these cells from a patient. Actually, this is a 14-year-old boy, and it's a sporadic mutation that happened just for this patient. But the point to follow up on Michael's explanation, before this is the best scenario for us as a proof of concept to show that our re-engineering, when we move them to the nucleus, these proteins are translated and they go to the right place. It would be very difficult for us to take a completely row zero cell line that's completely devoid of mitochondrial DNA and kind of proof for all 13 proteins at a time. They are all of very different sizes. They have high hydrophobicity, and most likely many of the genes will require additional engineering before we can achieve that. So the best condition for us to show is a completely null model where one gene is disrupted, and then we can engineer and we can bring back the function and we can show. And the hope is that we can extend the strategy to all the other 13 genes, and then we will be in a position to test in a situation where it's devoid of mitochondrial DNA completely. So this patient itself, the patient is unable to practically synthesize any ATP. The side effect is that he has severe cardiovascular impairment, skeletal muscle impairment. They are wheelchair bound, and it's just a severe phenotype. That's what it is. But there are other point mutations within the mitochondrial DNA, and there are several disease phenotypes like Leigh syndrome, LON, that is Leber's hereditary optic neuropathy. It affects vital functions like eyesight and hearing, and skeletal muscle dysfunction. There's also the Keir-Sire syndrome, that is again a larger deletion of the mitochondrial DNA, which is manifested in many humans. So our idea is, if we can show recapitulation of function in these patient populations, we can slowly extend our engineering, and we can achieve for all the 13 genes.
Right. And in that paper,
if I may, Oliver, just for a second.
Sorry, Michael, you're breaking up there for me. But you're mentioning a clinical condition where something's in the pipeline for treatment regarding mitochondria. I think that's where we last kind of got cut off.
Yeah, I don't know if you guys heard that because these various patient populations do have these specific mutations, unlike in aging, a quick route to the clinic potentially is to use specific allotopically expressed genes to correct the mutations with these patients. And one particular kind of allotopic, although we don't think it's a very, currently in clinical trials, there's two groups for a form of mitochondrial disease blindness, that they're using the ND4 gene to correct that deficiency and potentially correct that blindness. And the eye is also immune privileged, so it's a pretty safe place.
Okay, so I'm going to jump a little bit ahead. Obviously, when people hear about the progress that's being made on the research front, the question for everybody, including myself, is how do you actually take this proof of concept and translate it to a useful therapeutic, right? So it's one thing to have cells in a dish where you can basically weed out the ones that you want, pick the transgenic lines, show proof of principle that it works. It's a ton of work, to get to that stage. But then if somebody is already an adult, somebody is aged, right, which is now you have mitochondria affecting every single cell of your body. Perhaps some cell types are more affected than others. How do you translate this? And is there maybe kind of some thought given to how you would then translate this proof of concept into a therapeutic? So maybe somebody would want to chime in on how this could be moved in that direction.
Let me speak to this a little bit. We've given this a good amount of thought, and I don't know that we have a comprehensive plan for how we're going to get to the aged population yet, but I think there are a number of steps to take before we get there. We want to do experiments, as you said, in animal models first. The first one that we're aiming to do is in an animal with a mutation in the ATP8 gene that I think Kamutha may have mentioned earlier, or maybe she mentioned before in sort of the pre-prep for this, that will be the focus of our fundraising attempt. That will be a transgenic animal, so we're going to try to rescue the mutation in the animal with a transgenic approach. Another way to tackle that would be a little more realistic but less black and white in terms of results, would be to do gene therapy on the animal, which is undoubtedly the technology that will need to be developed in order to take this into humans. Now, we in our group, we're not gene therapy experts in terms of the application of gene therapy technology. So far, our strategy has been to leave the perfection of gene therapy technology to the experts, of whom there are many, many in the world, and simply develop the core technology for targeting mitochondrial genes to the mitochondria from the nucleus after having been re-engineered in the ways that Amutha articulated earlier. And so first we want to take it to animal models. Next, as I think somebody mentioned earlier, we want to take it into patients who have mutations in some of these genes in using technologies that are available or become available. So as Michael was saying, there's diseases of the eye, like Leber's disease, that people are trying to tackle. But there are other much more severe diseases that I think we're more excited about going after next, that we think will be more applicable to aging. So many mitochondrial deficiencies affect the neurological system, the skeletal muscular system, heart, and those are the ones that, as in our first manuscript that Mutha was describing earlier, that have the most debilitating and devastating effect on the patients. And we believe also the ones that are having the biggest effects in aging. And so I think that possibly we don't have any plans to move into clinical trials as of yet. But I think if our animal trials go well, that could bode well for trying to get our technologies into some, hopefully into some sort of clinical trial that's targeting something out of a simple immunoprivileged site like the eye into something more that causes more morbidity like muscle or the nervous system. So I think that's sort of the order that you'll see things roll out, and that things will start getting really exciting when we can start getting this into patients that are suffering the really debilitating effects of mitochondrial disease, and that will translate much more rapidly into aging. But as Michael pointed out earlier, we can't just do this with one or two genes. We're going to need to do all of them. So we've still got a ways to go.
Right. So yeah, that is one of the challenges, right, that we see with a lot of genetic defects, is how exactly do you target all of the cells, and clearly going to a model where you have trying to get localized expression, is a lot easier than trying to get a transgene to every single cell. The easiest way to do that is to start with a totipotent cell, but at that point we're getting into ethical considerations of engineering better humans from the ground up. Certainly, that's a topic for a future discussion, perhaps much later. But going back to the actual animal models and going back to the technology itself, the last update we had on our Leap website mentioned yet I guess here it's written about as a new trick that you have discovered improved the allotopic expression of mitochondrial genes. Can you tell us a little bit more about that? So an improvement in the technology that perhaps will be carried over into the transgenic mouse model?
Well, sure, I can jump in. We don't have all the details yet. We're still trying to make sure that we've established everything that we want to be able to prove in order to subject our results to peer review. But we've done a lot of work beyond the 2016 paper on this, in terms of trying to take this technology and generalize it to all 13 genes. And we have versions of all 13 of the genes that we've created now, and we've tested all of them in cells and gotten different ones to work to different extents. So there's a lot that's been discussed in the literature before about upstream and downstream elements in the RNA encoding components. that we are playing with in order to improve the efficiency, and a lot of things that we're working with to improve the overall expression of the genes, and also engineering like where they're being expressed from the genome. So we don't yet have a simple platform that we can apply to any of the 13 and have them work, unfortunately. So in some ways we're finding little tricks that may help all of them, and other things that we're having to focus more on individual genes in order to figure out why that particular gene needs a little more help getting either expressed or being targeted to the mitochondria, either at the RNA level or at the protein level, and then being imported into the mitochondria, and then being imported into the right place in the mitochondria. So there's different aspects of the mitochondrial targeting sequence, the RNA sequence, and the coding sequence itself that we're messing around with. And, I think we'll have something a little more coherent to say about that in in the next couple of months.
Okay, just backing up a little bit, or actually a lot, from therapeutics to aging all the way to evolutionary biology. And this is something that just kind of I've been thinking about a little bit with regards to your project and with regards to the kind of the flow of genetic information from mitochondria to nuclei, within the context of eukaryotic evolution. Is there a pattern here as far as which genes went first from mitochondria to the nucleus? Is this conserved between different species and different genuses? This flow of information, or is it just kind of, I would be surprised if it was kind of random and stochastic, process. Have you looked into that as to why certain sets of genes are in the nucleus versus other sets of genes which are still mitochondrially encoded? And is this vary between different species, or do we just see this conservation? Does this tell us anything about evolution?
Yeah, so in 2005, I published a comprehensive review of this topic that I believe is still the most recent comprehensive review. And it's quite surprising to me that nobody has kind of followed up on it. But the upshot, well, the essence of what I showed, what I concluded in that review, this was in bioessays, you can look it up easily enough, is that the process of transfer of mitochondrial gene coding mitochondrial genes to the nucleus has been constrained by two processes only, namely the hydrophobicity of the proteins that are encoded, which I mentioned earlier, and also the ability to compensate for genetic code differences. In other words, as Amita mentioned earlier, the fact that the mitochondrial DNA in modern eukaryotes uses a different translation system, whereby certain codons encode different amino acids in the mitochondria than what they do in the nucleus. Now, perhaps the most important items of evidence for this are, number one, that the original situation when mitochondria came into existence, what's called the endosymbiotic event, happened at a time in which the mitochondrial genome, as it then became, was actually the same in terms of codon usage as the nucleus. Most bacteria actually also have the same codon usage today. So that meant that the disparity of codon usage didn't exist. It only came into existence about 1 billion years later, in other words, about 1 billion years ago. It appears to have done so more than once, interestingly, but for sure, it's a big deal. Once that happens, and especially because of the particular change that happened first, which was where a codon that normally encodes the end of the protein became coding for an amino acid, we ended up in a situation where basically the transfer of genes from the mitochondrion to the nucleus just couldn't happen anymore. The barrier was elevated by a huge factor. essentially because the transferred protein would the transferred gene would now encode a truncated protein. So that's a really important thing, and that's the best explanation for why absolutely nothing has happened, as far as we can tell, in any lineage except one particular case called arrow worms. Nothing has happened at all in the context of genetic code disparity. However, there's a second thing which we can look at, which is plants, because it turns out that in plants, the genetic code has not drifted in mitochondria. We still do have the same genetic code in the mitochondrial DNA as in the nucleus. No codon encodes anything different, except, well, there is some ambiguities around start codons, but let's not go there. And there, the most important and informative group of species is blue-green algae, chlamydomonads. The most famous chlamydomonad, Chlamydomonas reinhardtii, actually has successfully transferred nearly half of the genes that remain in the mitochondrial DNA in animals. And that's a big deal, that's a lot of genes. So, of course, this is actually something that we ought to be studying. And actually, the first time that I started making a serious nuisance of myself among academics was back in the late 90s, when I pointed out that it was a bit of an embarrassment that no one had actually gone and sequenced the nuclear versions of these genes in Chlamydomonas, even though the mitochondrial genome had been sequenced back in 1990, and therefore it was known that the genes must have been transferred. So why is this important? Well, a really important aspect of it is that there's a closely related species whose name actually escapes me right now that looks almost exactly like animal mitochondrial DNA. In other words, the genes that the mitochondrial DNA retains are almost exactly the same, I think just one difference. That says, to me anyway, that the influence of hydrophobicity on transferability is enormously steep. In other words, that proteins that we might consider to be only slightly more hydrophobic by the measures that we have today are actually orders of magnitude, or at least one order of magnitude, more difficult to transfer during evolution. So this led me to the firm conclusion, which was actually the reason I wrote the paper, was because this was being challenged by people who were not paying enough attention to detail, that the hydrophobicity constraint and the genetic code constraint were the only two things that really mattered, at least in respect of animals in which there has clearly been strong selective pressure to move genes into the nucleus whenever you can. There's evidently been less selective pressure of that kind in plants, because plants tend to retain quite a lot of additional genes in the mitochondrial DNA, including ones that are not even particularly hydrophobic.
So kind of a follow-up kind of question to everything that you just said right now, and maybe this is kind of would be an overly simplistic hypothesis, but I guess based on the direction that the MitoSENS project is going, and I guess the hypothesis underlying it, and the fact that there is this difference in eukaryotes, some of them do have additional genes within the nucleus from the mitochondria. One prediction, obviously, would be that these cells that have more mitochondrial genes in their nucleus are now more tolerant to stress, or more tolerant to oxidative damage originating from mitochondria. Is this these experiments have been done? Is this something that's been looked at for naturally occurring species?
It's a great theory, but no, it doesn't really stand up to scrutiny. There are two problems with it. The first problem is that we're talking about plants here, and plants don't do so much oxphos in the first place. They're not warm-blooded, things like that. So it's not such an issue for them. But the other thing that's rather more important comes back to something that Michael mentioned earlier, namely, yes, you've got the right paper now, by the way, namely that in animals, the overwhelming majority of mutations that accumulate are mutations that knock out the entire 13 genes, because they knock out tRNA genes. And in plants, I'm not sure it's really been studied whether clonal expansion even occurs, let alone what kinds of mutations clonally expand. But I would say that this means that the difference between the two scenarios is so enormous that one can't really extrapolate from one to the other. And now you're muted.
Yeah, Fair enough. Okay, so kind of going now from evolutionary biology to back to clinical biology, and this kind of, I guess there's two kind of questions I have. One is obviously that one was that we sort of touched upon and we covered is how do we then go into a kind of a therapeutic manifestation of this. But I guess kind of the bigger question, and this is something that's been, we've talked about this for a long time, and there are ways around it. But how can a therapy like this eventually kind of get through or punch through the, let's say, the FDA wall if aging isn't being recognized as a disease? Now, of course, we all have our own personal answers to how this could be circumvented. But any thoughts on this? I know it's a really big question, but anybody want to chime in on this? Because there's hurdles to get things into the market, that's for sure. But anti-aging therapeutics, have their own peculiar set of issues to overcome. Thoughts, comments?
So this is what I was talking about earlier. The fact that there are these patients that exist with these individual mitochondrial mutations means that you can do clinical trials, clinical testing of the individual genes in patients that have that specific mutation and show individually that they're therapeutically effective and safe and everything else. And that just makes the case for putting them in aging people. Once you say, well, Parkinson's disease, there's these close associations, or sarcopenia or whatever, those are age-related diseases as well, where they can potentially be translated. And of course, over time, we expect FDA is going to become more and more open to this as aging becomes, increasingly targeted through other rejuvenation biotechnologies. And as FDA is, starting with things like the pain trial, starting to open up with aging per se as an indication. But the main thing is, you can go after specific diseases and then age-related diseases and then aging itself. It's a bootstrapping process.
Any other comments on this kind of weighty question here? No. Well, certainly that's a huge topic of discussion. We could have another webinar just based on anti-aging therapeutics and the concept of anti-aging and the concept of enhanced longevity and how how the future is going to bode for these technologies. Obviously, most of us here, myself included, are pretty optimistic about it. And there's a lot of hurdles to be overcome scientifically, but unfortunately, there's also hurdles to be overcome, politically and medically, and in entrenched establishments and entrenched ways of thinking. So I won't really go into kind of into that vein. So we covered a lot of different things here. I might have missed a few, but I think we pretty much covered a lot of different topics related to this project. We're about an hour in. I think this is a good time to, I guess, open up the mic to audience questions and anybody else who has questions to kind of, and I think some of them have been accumulating here. Steve, how do I do this? Should I just go to the message board and should we just read off of it? How do we get the audience questions kind of piped into everybody here?
Okay, well, the Q&A, if anybody does have a question, all they need to do is look at the bottom bar, and there is a hand icon on the bottom. And if you've got a question, just raise your hand, and then what we can do is then unmute you, and you can ask your questions. I hope some people have got some questions, because I've got a couple, but I don't want to be greedy and jump in first. So, yeah, if anybody's got a question, and you can actually work out how to raise hand. It took me a little while to work that out as well.
Can I raise my hand? Because I can't seem to, I don't see a little hand back.
You don't actually have that. Anybody who's a host or a co-host or whatnot doesn't have it.
Okay, I'm going to mute myself now.
And you have the power. Okay. Right, I'm just having a look. Okay. Well, I mean, okay. Well, I can go first if everybody doesn't mind. I don't see anybody asking the question yet.
Dean Key has a question. He doesn't have a hand, but he wants to ask one.
Oh, he doesn't have a hand. Oh, okay. You can also write in the chat if you've got a question as well, because the hand doesn't always work, and it might not work quite the same on mobiles.
He doesn't have a question either.
Oh, he doesn't have a question. I need to put my glasses on. Could
I
I
stand
volunteer?
corrected. I stand corrected.
Ah. Okay. He doesn't have a question anyway. I read that as I do have a question, so clearly I need some MitoSENS therapy, possibly some other therapies to help my vision. So hopefully we can get that rolling. Oh, okay. So, okay. Oh, I've got a question from Patrick. Patrick's one of our volunteers, and he's got a question. I'm just going to unmute Patrick. And Patrick, go ahead.
Hello. So repeatedly, SENS have said that once therapies have been developed, they'll be spun off into separate companies. I'm just sort of been curious about how that works. Like, how do you start off the spin-off, so to speak?
Let me take that one. So actually, there has been no one pattern there. What has happened has been rather different from one example to another. In some cases, it's been where a person who was working for us decided they wanted to start their own company to take things forward, and they felt that they could get investments, and so we just let them do that, and we transferred the intellectual property to them, and we would take a small stake in the company, an equity stake, but basically they would just get on with it. That's happened a couple of times now. One of the best examples is ICOR, which is run by Kelsey Moody. Then another pattern is when a professor that we have been funding for a while, purely philanthropically, decides to do this. In other words, to create a company. That's happened a couple of times also, and in most cases what tends to happen is that the professor actually quits his academic position in favor of being full-time at the startup. Another pattern is when a donor comes to who's been coming to us and funding stuff decides that the thing they're funding is ready for prime time and they take it private. All of those things have happened. And we're perfectly happy to be open-minded about this. We want these things to be well funded, and for sure it's a hell of a lot easier to get a hell of a lot more money into a project when the project is being pursued in a startup than when it is being pursued in the foundation. So this works extremely well for us. We are continuing to pursue this. Of course, the foundation remains indispensable as the kind of engine room of all of this, because we still have to do the work for as long as it takes to get something to a point where an investor will actually see that there is a value proposition there. And MitoSENS, of course, is a primary example of a project that has not quite got to that point yet, though we are definitely seeing the light at the end of that tunnel right now.
Oh, hopefully that answered the question for you, Patrick. I've got a technical, a more sort of biology question. I saw recently. I'm assuming I didn't imagine it. It's always been thought or suggested that mitochondria you inherit from your mother, but I saw a paper, I'm sure, last month that suggested that in some cases the mitochondria of your father may also be passed down. What do we think about that? Is that plausible or?
Yeah.
Okay.
Okay. Yes. Go ahead.
No, go ahead.
So when there is a fusion between the egg and the sperm, in the sperm, the mitochondria is concentrated around the tail, like more than 99% of the mitochondria, because for the motility, most of the energy is derived from the mitochondria, mitochondria in the sperm. And but when fertilization happens, this tail is discarded. So by theory, there is the first elimination of all the mitochondria that comes from the male. So for a fertilized zygote, most of the mitochondria comes from the mother. But there are, like you say, there are one or two reports here. There is a previous report too that says that the mitochondrial DNA can be paternal too. So they are anomalies, but for the most part, you inherit your mitochondrial DNA only from the mother. That's the scientific.
Oh, that's disappointing. I was, but then again, I was always told I had my mother's mitochondria, so I suppose nothing changes there then. Okay, we've got a question. We've got a few questions in the chat, and we have Sven, who looks like he wants to ask a question. So I'm just going to hand it over to Sven. Go ahead.
Hello, you can hear me? Okay, so you are familiar, of course, with the work of Carla Kruger from UCLA, who in 2012 published a paper showing that using PNPAs and highly modified RNAs, she could have those imported and have the translation of mitochondrial proteins done in the mitochondria itself. And I wonder, because I haven't kept up with her work, what is the latest update on her work in that regard? And is it very promising?
We don't have a lot to say on that. So far, we have been unable to repeat their results in our group. And I haven't seen any follow-ups. Maybe somebody, Muther Aubrey, might remember better than me. There's a group that may have shown something similar to that in fruit flies recently. I'm forgetting the details, but we tried it and couldn't really get that same kind of technology to work on a couple of our genes that we tried it on.
Yeah, I think, okay, the group that you're remembering there is the group from Pittsburgh of Michael Palladino. The work on RNA import is definitely something we continue to pay attention to. And there are a few other groups, including ones in Europe, that have been pursuing it for quite some time. But as you say, it's looking pretty dicey. There are some groups that have come to the conclusion that basically RNA molecules longer than about like the size of a tRNA are pretty much impossible to import for for unclear reasons. But ultimately, the difficulty is that even though some RNAs are naturally imported into mitochondria in many species, nevertheless, there are only a few that are, as opposed to 1,000 or more proteins that are imported. And that means that the machinery that has evolved to import RNAs is much more case by case. It's much more, difficult to generalize, whereas the protein import machinery is inherently versatile and therefore much more practical as something that we could co-opt for additional substrates.
Okay, we have a question from Alexandru. This person wants to know if there are any differences in causing mutations to mitochondrial genes between burning carbohydrates and burning fat ketones.
If I can take this, so there was, first off, some theoretical reason to think that burning fat might produce fewer raw from the mitochondria just based on the way it gets shuttled into the electron transport chain. And then in terms of ketones specifically, there were some short-term studies that seemed to show that animals fed ketogenic diets were producing fewer mitochondrial ROS, mostly because of a compensatory antioxidant response, which might or might not really have a strong impact on the production of these mitochondrial mutations. That is what we're really worried about, not so much the free radicals themselves, but the hitting of the mitochondrial DNA. But that was a relatively short-term study. There's been a much more recent study that showed that there are some significant impairments over the long term, like 8 months as opposed to a couple of weeks, when animals are maintained on a ketogenic diet as versus an isocaloric Western diet. That's another important thing. Whenever you have one of these studies, you want to make sure that you're not imposing accidental calorie restriction on the animals just because they don't like the diet, or it causes them satiation or energy loss through the fecal route or whatever. So that certainly suggests that a ketogenic diet over the long term is not going to be favorable to mitochondrial function and possibly to the actual deletion status of the mitochondria. Now, some people out there may be familiar with, there was a couple of studies, one by Eric Verdon's group at the Buck, and another one by a group up at University of California, Davis, that were the first really well done lifespan studies that seemed to show some quite favorable, even CR-like effects from a ketogenic diet, particularly the UC Davis group. Looked like a quite impressive effect, whereas Eric Verdon's results seemed quite mild. The unfortunate confounder to that study, when you dig into the details, is that as part of tweaking the diets, they didn't just adjust the carbohydrate and the fat. They reduced the protein intake, and for whatever reason that I asked them, and I wasn't able to get an answer that I found satisfactory, they wound up substantially reducing not just the protein as a whole, but the intake of methionine and cysteine in particular. And some of you folks may be familiar with the fact that substantial methionine restriction is itself an effective way to retard the aging process and extend lifespan in rodents. So, although it looks like there's this quite significant lifespan effect of the ketogenic diet, Occam's razor would suggest that what you're actually seeing is just a replication of the known finding that methionine restriction extends lifespan with, maybe no effect of the ketogenic diet at all, or for all we know, even, a deleterious effect of the ketogenic diet in as much as it is ketogenic, as opposed to in as much as it is a methionine restricted diet. So, in sum, I would say if you're in this for life extension, probably a ketogenic diet is not a good idea.
Interesting. We see people trying all sorts of dietary measures. Caloric restriction also seems quite popular as well. Nicola from Leaf has a question. You have the number one.
Right, thank you. So basically my understanding is that MitoSENS, the idea behind MitoSENS is that you will change the genome of a patient by incorporating mitochondrial genes into the nuclear DNA. But my question is, over time, it might be possible that nuclear DNA will mutate and the changes that you have introduced will be lost. So at that point, you would have to reapply the therapy, I guess. And my question is, first of all, is this correct? Is my guess correct? And second, if it is, then would you have to reassess whether the patient needs the therapy again, or could you safely apply it again every so many years without, even though, imagine the patient didn't really accumulate any or many mutations at all, could you apply the therapy again safely? That's the question.
Okay, or Emily, do you want to take that?
Okay, I'll try. We discussed about the difficulties in translating to patients, but the thinking is that with advances in gene therapy, the introduction of mitochondrial DNA would follow the same route. Like, as gene therapy becomes more and more safe, we will be able to introduce these mitochondrial DNA into the nuclear genome for either, severe mitochondrial dysfunction in patients or from an age related point of view. The thing is, the first premise that we want to transfer this mitochondrial DNA to the nucleus is that they have a better repair machinery. So you're thinking that this foreign gene, when practically a foreign gene that goes into the nuclear genome, if it mutates, do you have to reapply this? Then it would follow the same rules for any other gene that's in the nuclear DNA. But the the premise is that, okay, the nuclear DNA repair machinery is very efficient, so it should be able to fix any of the mutations that go with age. And then, of course, it is, removed from the OXPHOS reactive oxygen stress that it's experiencing, right? It's in two different compartments. So you've already separated that. So you've removed that let's say, a negative factor, and in addition, you're giving a better repair mechanism as a whole. So,
and additionally, as a third factor, you don't get the problem of these large deletions in the mitochondria as you do in the nucleus. So, if you got a mutation, it would be, perhaps a very small mutation in only one mitochondrial gene, as opposed to a mutation that is going to cause the entire system to just completely shut down.
And just to piggyback on this, just to take it a little bit further, related to what Nicola, the question that you asked. And this is a question that actually a colleague of mine here asked in the lab, which is, you have one gene that you're putting into the nucleus versus having 1,000 mitochondria with 1,000 backup copies. And the argument that I'm hearing is that in the nucleus you have, let's say, one versus a few hundred, but it's better protected. Has anybody modeled this, based on what we know as far as mutation rates in mitochondria versus mutation rates within the nuclear genome, that we can sort of safely put kind of a range or a number and say, okay, well, here's a number. If we have one gene in the nucleus, it is better protected than 200 to 1,000 backup copies that are dispersed in mitochondria, or is it still unclear that we can make this sort of back of the table napkin calculation?
So there's two things there. The first of which is it is many thousands of times less frequent that you get mutations on any spot in the nuclear DNA than in the mitochondrial DNA. But again, I'll come back to this point. The mutations we're concerned about in the mitochondria are these large deletions. And once they happen, it doesn't matter which specific genes it has knocked out or not. The mitochondria can no longer produce any of the genes from the ETS, and it clonally expands, takes over the entire cell. So the healthy mitochondria just get crowded out, and the entire cell is just down and toast. So whereas if you put them in the nucleus, you don't have any of those problems, right? You have, at worst, probably a minor mutation in a single mitochondrial gene that might introduce some level of dysfunction, but is not going to completely eliminate the ability of the mitochondria to function.
Okay, do we have any more questions, Steve, coming up that I may have missed from folks out there?
There is a question from Eric W. He is asking what the next likely gene candidate is going to be after ATP6 and ATP8. He wants to know if you're going to plan on putting the last of the 11 genes into categories or tiers. He wants to know if there's a roadmap for the whole 13 genes as to when things are going to be completed.
Okay, I can take this because I work in the lab and we have a very good team here. Yes, we are working on other genes. I want to say that at least one other gene is working very efficiently for us. Without giving too much away, we are putting together a manuscript, so I don't want to give away too much. But the ND1 gene is working good for us, and we are testing this again in another patient null cell line, and we have some very positive results. But again, you know, most likely some of the lesser hydrophobic genes are likely to work in our strategy, and there are proteins like cytochrome B or COX-1 that are extremely hydrophobic. Where when Aubrey explained that the Chlamydomonas strain, that it has transferred many of the genes that are present in the human mitochondrial DNA to the nucleus. Only 2 genes across all eukaryotes are still retained within the mitochondrial DNA. These are cytochrome B and COX-1. So most likely these 2 genes we have to do much more engineering from the efficiency perspective. But we are hoping that the rest of the genes, you know, they'll be, we have some tools up our sleeves to get them to work.
I'd like to expand on what Amutha just said, actually, because there's an important point here which leads us to even greater optimism that we are getting quite close to doing the full job. And that is that even though there are, as Amutha said, only 2 genes that are always found in any species in the mitochondrial DNA, there are more genes that are not found ever in the nuclear DNA. The reason that can be true is because those genes are in complex 1, and complex 1 has simply disappeared from some species, in particular from the 2 main species of yeast that are studied as model organisms. Now, this is really important because it tells us that actually those subunits of complex 1 that are never found in the nucleus may be as difficult to transfer as the 2 that are always found in the mitochondria, cytochrome B and COX-1. So that's quite good news, because one, as Amutha just mentioned, some of the genes that we are now having considerable success with are subunits of complex 1. Indeed, ND1 is one of those 4 that are never found in the nucleus. So actually we have a considerable basis for optimism. Go on with that.
Steve, you're about to say something?
Oh no, somebody has a hand raised up there if they want to ask a question.
Okay.
I was about to get that. That's Victor Björk. You have a question? Please go ahead.
Okay, can you hear me? Yeah. Yep. I was thinking about the Pumilio 2. I mean, there have been some recent studies showing that if you can inhibit it, you can get better mitochondrial function. You are increasing mitophagy, for example. What do you think about this?
So I missed the very beginning of that question, Victor, but in terms of mitophagy, this gets back to this problem of clonal expansion. There are there's a little bit of debate as to what exactly causes this clonal expansion problem in the mitochondria. But the model that we favor, that is Dr. de Grey's main claim to fame historically, is based on the fact that there is that mitophagy in a sense gets subverted when you get these large deletions. That when you get a large deletion in a mitochondria, it acts as a kind of a cloaking device whereby the mitophagy machinery can no longer see that mitochondrion. And that's why they survive these rounds of mitophagy and actually wind up expanding, because the healthy ones get culled and they just keep growing and growing and growing and growing every time there's a round of mitochondrial replication. And certainly in the years since Dr. de Grey published that as his thesis, there's been a series of papers that certainly do show that there is a triggering of increased mitophagy in the very early stages of clonal expansion, and that there's a correlation between those two events. That is, the evidence seems to suggest that Dr. de Grey is correct, that indeed stimulating mitophagy in these specific cells, as opposed to other cells where you might have more minor defects, is actually contributing to bringing the process to a head and causing the cell to just get completely buggered up. So I'm not going to say that, any particular supplement or lifestyle practice or whatever therapy you want to pick that stimulates mitophagy is going to cause crippling increases in mitochondrial mutations. But clearly, at the very least, if that's correct, it's not going to help.
Well, I hope that answered that one, Victor.
Yeah, but I was thinking about this protein Pumilio-2 that you maybe, because it was recently published, if you have.
Yeah, Dr. de Grey, do you want to take that one?
Well, actually, to be honest, no. I think that at this point, the evidence from that paper and from related work is still too preliminary to allow us to persuade us anyway to alter our priorities in terms of research. I think, we'll be watching that work and related work closely as things go forward, but at the moment, it's not clear.
It's a good answer. It's a good answer. If anybody's got any more questions, keep them coming. Yeah. I wanted to ask a more general question. Of all the 7 deadly SENS, as some people call them, do you think that MitoSENS is the most challenging of them all, or not? Which would you agree it's one of the?
So let me take this one because it is a question that I keep very much at the front of my mind over time. The reason I do so, of course, is because as Chief Science Officer of the Foundation, I have overall responsibility for identifying our priorities in the Foundation. And what we've always had as a strategy, as a choice, as a metric for identifying those priorities is the difficulty of the problem. We understand very clearly that SENS is a divide and conquer strategy, and therefore that we need to get everything working. And as such, it makes sense for us to prioritize the things that are the most difficult because they need to catch up. This is, of course, even more the case when we take into account the fact that other researchers who are constrained by peer review and such like tend to be biased in favor of working on the low-hanging fruit. So the things that are most difficult get even more neglected. All right, so then coming to the question, well, historically what I have said is that actually oncosense is the hardest. That cancer is the most difficult part of aging to fix. I even said this back in the very first SENS paper back in 2002. And the reason I said that, and the reason I have historically thought that, is simply because cancer is the one aspect of aging that has natural selection on its side, where it can get cleverer as the attacks on it get cleverer. And that has been my view until quite recently, but now I'm a little less sure largely because of the breakthroughs that have occurred in cancer immunotherapy over the past 5 or 6 years. And I'm not just talking about the breakthroughs that everyone knows about, like CAR-T and checkpoint inhibitors. I'm talking about kind of second-generation things that are not necessarily on everyone's radar yet, but are very much coming and are exploiting our ability to do really high-throughput omics on people's cancers so as to personalize things well. There's a huge amount going on there, and it's very well funded. One of the things that I am really happy about is that Sean Parker, who, of course, is a wealthy guy and very visionary, co-founded PayPal and all that. He's Facebook. He's a guy that I met a long time ago and tried unsuccessfully to recruit as a major financial supporter. But now he is putting very large amounts of money into cancer immunotherapy, and I'm really delighted about that. So, as far as I'm concerned, he's doing my job. But why I mention all this is that it means that we may not need in the future to view cancer as the number one problem. Many of you will know that the SENS approach to cancer, namely wilt, is really ambitious. I mean, really, really, really difficult. And in fact, there are plenty of biologists out there who think that the rest of SENS is quite reasonable, but that wilt is just completely infeasible. So, it's worth mentioning that wilt may not be necessary. And no one would be happier than myself if that turned out to be true. So where this leaves us, coming back to the original question, is, of course, what else is really hard? And I would certainly say that MitoSENS is, at this point, definitely the one that competes with cancer, with OncoSENS, as the most difficult. All of the other areas of SENS at this point have moved forward, at least in regard to certain examples, into startups, and in most cases into clinical trials. So we're looking now at a situation where really OncoSENS and MitoSENS are the two things that need the most work. Even GlycoSENS, the elimination of crosslinks, is now being pursued with a company that has been spun out from our group that we funded for several years at Yale. So yeah, I would say MitoSENS is really up there as the thing we should be focusing on.
Right, thank you very much. Oliver, you wanted to jump in?
Yeah, nobody has, anybody else has a question, feel free to jump in. I'm just gonna, chime in a little bit since we are talking about mitochondria and we are talking about, ways to ameliorate the damage that's happening in mitochondria. Clearly, one approach is to defend the mitochondrial genome against onslaught. Another approach that has been in the papers for quite some time is to mop up the free radicals themselves, through the use of antioxidants. There's one in particular that's been published and is, I believe you can purchase it, MitoQ, which is a derivative or a version of a synthetic version, a variant that's of coenzyme Q that targets it to the mitochondria. And my question is to the researchers, Aubrey and Oki and everybody else here on the panel, what do you think about those approaches? And do you think those approaches would supplement or be additive to the approaches that you are considering as far as, protecting, defending the mitochondria from damage, since it is such a crucial element of the cell and the role it plays in aging? So, thoughts, comments on antioxidants, particularly things like MitoQ. And
so I can take this generally. I don't want to get into too many specifics about any individual mitochondrially targeted antioxidant because I'm likely to misremember the details about what the particular problems with particular mitochondrial antioxidants are, because there's several of them. There's MitoQ, there's a peptide called SS31. there is this peptide produced by a Russian named Skulichev. They all show some advantages you might think would occur in disease models of different kinds, but whenever they've actually been tested in normally aging animals, they either show no benefit or in some cases mild deficits. MitoQ in particular, there's been some unpublished work by a guy named Navdeep Chandel showing some potential problems with MitoQ. And certainly it hasn't been shown to have any real advantages in a normally aging animal. The most likely reason for that is that, they're not really effective, even though they're supposedly mitochondrially targeted, they're not really effective in actually offloading the free radicals that are produced, even if they manage to intercept a given, lonely electron. And also just stoichiometrically, you just can't pack enough of these things into a close enough proximity that the mitochondrial DNA is not the easier target for most of them to hit. So I would say save your money and either invest it in SENS Foundation or into whatever entertainment purposes fit your mind, because it's not going to be any good.
Okay, thank you very much for that. We have a question, I believe, from Stryker to everyone. How much do you think damaged mitochondria's damage affects exacerbate other areas of the body, given how they cause stresses throughout the body? I.e., could conquering MitoSENS be the best bang for the buck in terms of longevity? I guess that gets down to the, to the heart, to the root of mitochondria really being the kind of the powerhouse of the cell. So
yeah, let me take that. Let me take that, because this is something where my own view has actually evolved a little bit over the past few years. The original situation, as of, let's say, 10 years ago, was that MitoSENS was a bit of an orphan, a bit of a Cinderella with regard to SENS, in the sense that all the other 6 aspects of SENS, the type of damage that they define, could be very clearly and explicitly associated with a particular pathology late in life that humans actually experience. Something that, would be a pathology whose main cause was pretty clearly this or that aspect of SENS. Whereas there was no aspect of, no pathology of aging for which mitochondrial mutation accumulation was clearly the main cause. And so the thinking was clearly that the way in which mitochondrial mutations matter is very much more diffuse. Essentially, that there is an indirect but very fanning out, broad-based mechanistic basis for the influence of mitochondrial mutations on old age, ill health. And as such, it was hard to say whether mitochondrial mutations really would be the major player, or maybe they would just be a rather minor player that would just mildly accelerate things that were being driven by other aspects of SENS. But now my thinking on this has somewhat changed, and it's largely changed because of what we have seen with senolysis, with the elimination of senescent cells. I think that even people like Judy Campisi would have said a decade ago that probably the elimination of senescent cells would have specific effects. It would have benefits in certain tissues that accumulated a lot of senescent cells, like, for example, cartilage, or maybe in ones in which the secretory phenotype had a particularly powerful impact on, for example, the extracellular matrix. I think even people working in the field and people who pioneered the idea of a SASP, Judy in particular, would say, as of today, that they would never have expected the sheer magnitude of and breadth of the impact of senolysis on overall age-related physiology. It's been extraordinarily large. And what this says really is that the magnitude of the crosstalk between different components of SENS, between different types of damage, may have been underestimated, may be greater than we think. This is fantastic news in a whole bunch of ways. The first fantastic aspect, of course, is that it is it's an indication that we may be able to get to longevity escape velocity with a more imperfect, a more incomplete panel of interventions than we might previously have thought. In other words, that the crosstalk may essentially compensate for things that lag behind, for aspects of SENS that we are slowest to actually implement. But of course, the other thing is that it may also be true for other aspects of SENS, same as it seems to have been true for senolysis. And that tells me that maybe we should be expecting that allotropic expression, once we do it thoroughly, will have a bigger impact on the rate of progress of other aspects of SENS than we might have estimated 10 years ago. Should I be saying over?
Very good. Over and out. So do we have any other questions from anybody out here? I'm looking at got one eye on the chat. I don't see any questions popping up. Anyone? Mic is being passed around.
It's because it's such a super hard topic, mitochondria.
Yeah, well, also we covered a lot. I think we basically ranged from evolutionary biology to therapeutics to tackling the genome to other approaches to mop up or potentially not mop up, free radicals to the minutia of how to get the genes into the nucleus. We really covered quite a lot of ground, I think, in this webinar here. And if there are some questions that we have not covered, then they're really escaping me right now, because I think we touched on a lot. Obviously, we could range even further across the entire field of longevity research and anti-aging therapeutics, but with regards to the MitoSENS project, I think we ranged pretty far and in some cases pretty deep. Let's see. Yeah.
There
we go.
Would MitoSENS help with a natural age-related drop in NAD plus concentration naturally? This is something I was thinking about asking because I'm quite interested in NAD plus biology. So it's a great question, that.
All right. Especially ties into a lot of other supplements that are on the market, and maybe additional supplements yet to be on the market.
So any thoughts on what Eric's asking there about would MitoSENS help the natural age-related drop in NAD?
So most likely the real problem in aging is not an absolute drop in NAD, but a shift in the NAD to NADH ratio. And the reason why these precursor supplements deliver the benefits that they seem to, at least in rodents, is that by dumping in a whole bunch more of the precursor and initially boosting up the NAD level, you are altering that ratio. Certainly through changing by eliminating the systemic oxidative stress effects of mitochondrial mutation-bearing cells, you ought to be able to have an impact on that. But I don't think we understand the reasons for the systemic decline in the NAD-NADH ratio, or even the possible decline in NAD levels total, well enough to say what exactly is driving that, and therefore what effects any individual rejuvenation biotechnology would have on it. We're just going to have to test them and see, basically. Yeah.
Very good. So open to discussion research-wise. How far do we want to range here, Steve, with our questions? We have 15 more minutes left scheduled on our webinar here.
Yeah, I mean, maybe a few general SENS research questions, if that's all right. Because I'm sure people want to talk about the cool topic right now, which is senolytics. I mean, you know, if you're not interested in senolytics, you're not cool, apparently.
And maybe some of the questions that we can field right now would, with some judicious splicing and editing, is a nice segue to our next topic.
If you're not interested in senolytics, you're not awake, is what I would say.
Oh yes, I'd be wondering where you'd been for the last 2 years, to be honest, if you didn't know about senolytics. But yeah, that's definitely a hot topic. If anybody's got any general questions, please go ahead. Please let us know. Any general questions about SENS? Patrick has asked, now that glucosapane... Oh, hold on, we've got to double up. Now that glucosapane research is moving smoothly along to spin-offs, what's the next step for glycosens?
Yeah, I'll take that. I'll take that. So, as many of you probably know, we have been funding 2 glycosens projects for the past few years. In parallel with the work of David Spiegel's group at Yale that's being spun out into a company and pursuing glucosapane, we've also funded a group in the UK headed by a fantastic chemist named Jonathan Clark. And that work has been precisely to look for additional crosslinks that might also be substantial contributors to the loss of elasticity of various tissues, whether the skin or the major arteries or whatever. The reason for this is because even though there is an established consensus in the literature, and has been for a long time, that glucosapane is by far the dominant contributor to age-related increase in cross-linking and therefore to stiffening. Nevertheless, as with so much of science, this may be a premature conclusion. It may be that there was too much of, finding what you were looking for. And so we have felt that it was crucial to employ an extremely hypothesis-free and objective approach to the search for additional crosslinks that might have been overlooked just because in whatever way they were just too different or too easy to overlook. And so far, the results of the work in the UK have not turned up any particular red flags, any particular candidates, but we're not done yet. So we are definitely planning and expecting to continue that work for the foreseeable future. Essentially, you can regard it as de-risking of the work that's going on glucosapane. If glucosapane really is the major player, as it's believed to be, then all well and good. But if it's not, and we only find out that it's not when we get to clinical trials, then we won't be as far along as we would have liked. So this is an essential parallel research theme that may potentially throw up a new target that, of course, we would then pursue in the same way that we pursued glucosapane, identifying enzymes or small molecules that would break it.
We have a question from Chris Linnell. He's asking about with the success of the end of the year fundraiser, where do you plan to invest the extra funds and what projects do you plan to initiate?
Well, first of all, let's be clear that even though our end-of-year fundraiser was very successful by historical standards, it still was considerably less in terms of actual dollars than what we received a year ago at the time of the huge spike in cryptocurrency value. Remember, back then, a year ago, we received 4 major 7-digit donations, adding up in total to $6.5 million. And for sure, we have been aware that that was unlikely to happen again, and therefore that we need to be careful in how rapidly we spent it. The result with the past year has been that we've been quite cautious. We have funded one major new project in the Albert Einstein College of Medicine, looking at a new approach to using stem cells against Alzheimer's. And we've also expanded a number of our other programs, including our education program. But the fact is, we are still living to a certain extent off the money that came in a year ago. To answer your question more directly, though, we always have a wish list. We always have a kind of a list of things that we'd quite like to fund, whether things that we thought of or things that were suggested to us. And we revisit that wish list quite often. And of course, reorder it in the light of developments elsewhere, whether they be scientific developments that make something more likely to work or less likely to work, or whether they be funding developments, like, for example, an investor coming in and allowing a project to be funded commercially when we had previously thought that it would have to be funded philanthropically. That happened actually a couple of times in the past year. So the answer to your question is, we definitely can't answer the question, and that's how it ought to be. We want to be making our decisions in a very nimble way, in a way that is determined on the basis of recent discoveries, recent events, and we will make those decisions when we're ready.
Great answer. I've got a couple actually of questions, if nobody else minds. There are a couple of mouse questions. I'm a bit obsessed with mice, unfortunately. I think it's something to do with the MMTP project. I'm obsessed with mice. I wanted to ask, how is progress going with the maximal modifiable mouse? And one thing we do get asked quite a lot is about robust mouse rejuvenation, or RMR. How far away are we from that? Are we still on the 5-year-ish prediction, or?
Okay, you take the maximally modifiable mouse question, and then I'll take the RMR question.
Sure. So for the maximally modifiable mouse, we've just kind of rebooted this project in a way within the last year, and are working with a contract research organization that actually was co-founded by somebody who helped invent the technology. So we're working with the best partner there. Right now we're at proof of concept stage of trying to use the maximally modifiable mouse as a gene therapy model. So maybe I should take a little bit of a step back and talk about what the maximally modifiable mouse is for just a second. It's a mouse that we have engineered to put in an integrase. So we've engineered, so it's an artificial mouse. We've engineered it to have an integrase site in its genome at a well-characterized site where genes can be well expressed from. And this actually has a huge number of applications for both basic science research and, we think, for gene therapy research. So right now, it's already been well established that it's a superior gene therapy mouse. A superior transgenic mouse. So anybody who's making transgenic mice should be using this technology, but hardly anybody is. But that's actually available. You can go to this company, Applied Stem Cell, and get your transgenic mice made from them, and it costs a few $1,000s more, but they're just way better than everybody else's transgenic mice, so they should do it. The second part of that is that we want to be able to use this technology for gene therapy, and we're at the early stages of proof of concept of just trying to turn different organs of theirs green, and we're awaiting some our version 1.0 of that, where we're just systemically delivering DNA that should be able to get GFP, green fluorescent protein, that should be able to get integrated into the mouse. We're expecting that our version 1.0 of this is not going to be super efficient. We're using a very primitive technology to do it, but we just want to see if we can at least turn their livers green before we move on to some more sophisticated aspects of ways that we can use this mouse as a gene therapy model and maybe find ways to translate that into some really sophisticated human gene therapy technology in the future.
Yeah, thanks, Oki. Regarding your RMR, robust mouse rejuvenation, I think we're getting pretty close. I want to come back in relation to this to my previous answer regarding the role of MitoSENS in SENS overall and the role of senolysis in SENS overall. If I'm right that the magnitude of the crosstalk between different components of SENS is higher than I would have said a decade ago, then that means that both robust mouse rejuvenation and robust human rejuvenation are going to be easier because we will be able to reach those points with a more incomplete panel of interventions. Therefore, I would say that this is why I have actually reduced my estimate for how soon we have a 50-50 chance of getting to robust human rejuvenation from about 20 years to about 18 years as of literally the past year. In other words, we've genuinely saved something. But with regard to robust mouse rejuvenation, I think the same applies. We already know that mice can have their longevity extended considerably more than humans or long-lived other animals using very simple techniques such as calorie restriction. I think that we still need a lot more work on the evaluation of late onset interventions, things that are started at 18 months or 2 years of age, which of course is the definition I always gave to robust mouse rejuvenation. However, the fact is that more of that is now being done as well. For example, Brian Kennedy's group in Singapore, which is, I believe, going to be a really major player in our world over the coming years, they are doing most of their experiments with late onset interventions now. It has, the world seems to be finally waking up to the fact that, yes, you're more likely to get a positive result in a nice high-profile publication if you start something at weaning. But the fact is, if you want to find out something that actually has long-term real significance, you'd better start late.
Oh, that was a great response, thanks. And it's one reason why we used already old mice for our senolytics. At MMTP, as you know, we're testing combinations of senolytics, which is kind of in line with Peter de Keyser's thinking of senolytic cocktails, because they all hit different pro-survival pathways, and different senescent cells use different pathways, apparently. So we'll see what comes of that. That's great. Does anybody else have any questions? Anybody? Oliver seems to have stepped out, which is great. He's just gone to get a coffee. So I think that's about rounds it up. We're all going to be in Berlin, I believe, or at least most of us. I know. I'm not sure, Oki, are you going to be there? You're going to be there, aren't you?
Oh, absolutely. There's going to be a very strong showing from the SENS team. I think our entire MitoSENS team, including intern, are going to be there, and people from both of our other internal projects. I can't remember at last count. We were going to have something like 14 or 15 or something people from SENS at the Berlin conference. So if anybody's been on the fence about signing up for Undoing Aging in Berlin at the end of March, I highly recommend you doing it. It's going to be a phenomenal conference.
Yeah, absolutely agree. It's a testament to the hard work that everybody's put into it. Fantastic event. Enjoyed myself last time. And we're going to be out in force as well. There's going to be 6 of us at least, maybe 7. So I guess that's a lot of rounds in the pub afterwards as well. Sorry, I'm English. I couldn't help that. It's a cultural thing. But I'm not paying for lunch for everyone. I'm just going to let you know that straight away. Not that I'm,
And not to toot our own horn, but a few months after the wonderful Undoing Aging conference, which a lot of us are going to attend, LEAF is going to host its second annual conference here in New York City, where there will also be many rounds at a pub right across the street, McSorley's, the oldest operating bar in New York City. So that's going to take place in July.
Yeah,
so
that's 2 days this year as well, isn't it? 2 days.
It's going to be a 2-day event instead of just a 1-day event, since it was so successful last year. So that's 2 conferences I'm looking forward to.
Yeah, I'm looking forward to them all. The more, the better, really. You can't get enough aging research advocacy, especially those kind of events. So you can count me in, and I'm not just going for the beer, although that's a pretty good reason. So I think we're about wrapped up, aren't we, Oliver?
Yeah, I think so. 4 o'clock on the dot, 2 hours on the dot. I think we covered everything and more in the time that we had allotted. And I'd like to thank everybody from SENS who joined us, Samutha, Matthew, Aubrey, Michael, and everybody else who's been on the, either quietly lurking in the background or shooting questions forward. Thank you. Thanks to everybody for making this happen.
Yeah, and I shall see some of you in London, perhaps in a week's time. We will be there as well. We're everywhere, really. I don't know how we do it, but if anybody can clone some of us, that would be really helpful. So if you guys, got any contacts in that respect, we would appreciate a few of our clones, so we can have a bit of a break. And
yes. Send the human cloning emails to Steve, not
Steve,
me.
at noethics.com. But there we go. So we've got a lot to look forward to. There's a lot of events coming up, and hopefully we'll have some more news about MitoSENS soon. So we'll keep you posted. So stay tuned. And thanks to everybody for joining us today and making it possible. And a very big thank you to all our lifespan heroes as well for helping us do what we do. Without you, we couldn't do what we do.
Yeah, and thanks to everyone who contributed to the original fundraiser. That really helped us push that project over the finish line and got us going with the stuff we're going to be unveiling shortly.
Yeah. So let's see if we can double it last time. It would be great if we could. It would be great if we could hit 100,000. Maybe I'm just dreaming, but 100,000 for MitoSENS would be great. Let's give it a go.
Yeah, thank you very much. Thank you very much for hosting this. Yeah, it's exciting.
Yes, thank you, Steve and Oliver. This has been a really worthwhile webinar, and I'm sure it will be the first of many.
Yep. Right. Thanks everybody for making it happen.
Thanks very much, and we'll see you all soon.
I had a lot of fun. Thanks, guys.
Thanks, guys.